Hey guys, this video lecture is going to be over viruses, which is pretty much the last topic that we need to cover to finish off our curriculum. So here we go. So viruses don't really follow under our usual concept of life. A lot of biologists think of them as having a borrowed life, and it's because there's certain things that they really just can't do on their own. So just to give you an example, there are types of viruses called bacteriophages, and bacteriophages use bacteria, all kinds of bacteria, as their host cells. So what they do is they infect and pretty much genetically take over the bacteria, forcing it to make replicas of this virus. So for that reason, we kind of say that they have a bit of a borrowed life, because there are certain things they just can't do on their own. A lot of what we know from early molecular biology is all built on viruses that specifically affect bacteria. That's kind of where we first started developing the field of molecular biology. So in terms of structure, viruses are not cells. They're very small infectious particles that are made up of several parts and pieces. The most important parts and pieces would be the fact that they have genetic material in the form of nucleic acids. Now they can have either DNA or RNA, and their DNA or RNA come in two forms, double or single-stranded, so it just depends. Directly around the nucleic acid, they have a protein coat that is called a capsid. So if you're looking at this picture, this red part right here, that's the genetic material, and then these purple little, I don't know, they look like, almost like little Legos that you've built together, and now you have this tube. That's the capsid that surrounds the nucleic material, or the nucleic acid. In some cases, they will have a membranous envelope. So it's not really a cell membrane, it's just an envelope around their capsid and their RNA. And on this envelope, they'll have surface markers in the form of glycoproteins. It's like these little purple things that look like pins. Those would be the surface markers. Now, the viral envelope, this part right here, they actually derive it from their host cell's membrane. And that membrane is going to contain a combination of both viral molecules and host cell molecules. So in a lot of cases, having this membranous envelope makes it easier for that virus to either attach to a host cell or dock to a host cell. Here's another example of a virus. These are called bacteriophages, and they specifically affect bacteria. And they have one of the most complex capsids found around any of the viruses that we've discovered thus far. So the phages have a long, elongated capsid head, which is this region here. So the purple part on the outside that looks like triangles, that would be the capsid. And inside of their capsid is their DNA. So these blue squiggly lines, that would be their DNA. They also have a tail piece that's made up of protein, this region. And they use that to attach themselves to their host, and then they kind of inject their DNA into the host cell. Now, one of the reasons that viruses are not considered alive is because they can't replicate without the presence of a host cell. They are obligate intercellular parasites. They need a host. They can't exist without one as far as reproduction and replication occurs. So replication must take place within a host cell. So viruses do not make baby viruses or anything like that. They take over a cell and utilize that cell's DNA and enzymes to go through protein synthesis to make their parts and pieces. Each virus has what we call a host range. And a host range is a limited number of host cells that it can infect. So they may not infect just one kind of cell, but there'll only be a few kinds of cells that they can infect. So for example, they might infect liver cells and red blood cells, but that would be it. Okay, right here should have been a video that would go through how viruses replicate. we'll play this in class because it doesn't work under this particular medium. So here's a pictorial format of what that video would have shown, and it just shows how viruses will get their DNA or their RNA inside of their host cell. So the first step happens with what we call entry and encoding. The virus, so here's your virus, the blue part's the DNA, and the little purple balls are the capsid. The virus will dock onto the host cell, and it will insert or inject its DNA into the host cell. And sometimes even some of the proteins in its capsid will get injected into the host cell. That viral DNA is now going to become a part of this cell's chromosomes, the cell's DNA, and it will undergo replication, so more copies get made, and it will undergo protein synthesis, and we will make messenger RNA from it, and then through translation of protein synthesis, proteins would be made based on the genes on this piece of mRNA. So once we have nucleic acids, once we have viral material, and we have capsid proteins, we can start to reassemble smaller new viruses inside of that host cell. So each new virus will have a little bit of viral DNA and some capsid proteins. And that keeps happening until the cell can't hold anymore, and it bursts or lyses, and the new virus particles are allowed to exit the cell. That's pretty much how a virus will take over a cell and form new viruses using the host cell's machinery. Okay, so there are two stages that all viruses undergo, or can undergo. The first one is called the lytic cycle. In the lytic cycle, the phage, or the virus, is actively replicating, and it results in the death of the host cell. So this stage is going to produce new phages, and enough phages are going to be produced that eventually the host cell will lyse or break open, and the new viruses will be released out into the environment that they're in. Viruses can transmit DNA or RNA when they infect a host cell. So this would be transduction in bacteria. You're giving new viral information into the bacteria. And a phage that's made through this lytic cycle is what we call a virulent phage. One that's active, one that can infect another cell. Bacteria do have some defenses against phages, and we've talked about them before. When we talked about biotechnology, we talked about restriction enzymes. And their job is to recognize and cut up certain phage DNA based on restriction sites. All right. Another video that we will play in class, because again, not supported by the medium I'm using to make this video. Okay, so these are the steps within the lytic cycle, and it starts off with the attachment of the virus, then the entry of the DNA, and the degradation of the host DNA, and then the synthesis of viral genomes and proteins, the assembly of the new proteins with their DNA, the lysing of the cell, which allows the new viruses to be released. So that's cycle 1. Cycle 2 is called the lysogenic cycle. And the lysogenic cycle replicates the phage genome, but it doesn't destroy the host. And at this point in time, we're not actively making viral DNA or viral proteins. So the viral DNA molecule is actually incorporated or hidden within the cell's own chromosome. It's almost like the viral DNA becomes dormant for a bit. Every time the host cell divides, it's going to copy that viral DNA and pass those copies on to any daughter cells that cell division would produce. But in this case, the DNA is dormant. We're not actually making viruses, but every single cell that is made from the original, or that's reproduced, based on the original through mitosis, will have that DNA, that viral DNA, incorporated into it. So when we switch from the lysogenic cycle into the lytic cycle, all of those cells will be capable of making viruses. Alright, another video. Moving on, we'll play it in class. Alright, so what decides which cycle a virus does, lytic or lysogenic? It really does depend on the environment that viral genome exists in. That's going to determine whether you switch to the lytic cycle or whether you switch to the lysogenic cycle. So this just shows you, this particular picture just shows you that the two cycles are actually kind of intertwined, and you can go fairly easily from lytic to lysogenic or from lysogenic to lytic. Okay, viral envelopes. Now, this just shows how a virus will get its envelope. So you know we talked about the capsid. These are the proteins that surround the genetic material in the middle. When that cell lyses and those viral molecules pass through the cell membrane, sometimes they will wrap a portion of the cell membrane around their capsid. And that's how you end up with this viral envelope. This is actually fairly smart because when this virus chooses to infiltrate or attack another host cell, which is going to be whatever this cell originally was, it already has glycoproteins that these original host cells will recognize. Because it's like if this was a red blood cell, all of these surface receptors are going to be receptors found on a red blood cell. So if this virus went to attack another red blood cell, the new host wouldn't necessarily think anything's wrong because these are all proteins that it would recognize. So think of this as another form of protection for the virus. Okay, now viruses have the ability to use either DNA or RNA as their actual genetic material. And you see a lot of RNA genomes in viruses that infect animals. But at the end of the day, we still need DNA. So what these viruses have had to do is find a way to take RNA and turn it into DNA almost. And there are types of viruses that are very good at that. They're called retroviruses. And they use special enzymes called reverse transcriptase enzymes to copy their RNA genome into DNA. So if we think of it, reverse transcriptase. Transcription normally takes DNA and turns it into some form of RNA. But if we're reversing transcription, we are taking RNA and turning it into DNA. So that's what retroviruses do. A good example of a retrovirus would be HIV, the human immunodeficiency virus, which is the retrovirus that will cause the disease or condition AIDS, acquired immunodeficiency syndrome. Right, and this is just a picture that shows you that process. So here is your AIDS virus. What am I saying? Here's your HIV. Sorry. The RNA gets into the host cell. And the first thing that's going to happen is the reverse transcriptase is going to go through and make a copy of DNA from RNA. And then the DNA gets infiltrated into the chromosome of that actual cell. And then protein synthesis happens as normal. We make new HIVs and they leave the cell. So I've said before that viruses do not fit our definition of living organisms, mostly because they can only replicate within cells, which leads us to think that they probably evolved as bits of just cellular nucleic acid. They are candidates for the source of viral genomes are things like plasmids. So where is this actual, nucleic material coming from? It can come from plasmids, from circular DNA in bacteria and yeast, from regions of the genome called transposons that can actually move around, from small mobile sections of DNA. Yes, DNA can move. All of these pieces, plasmids, transposons, and viruses, are pretty much all mobile genetic elements. They can all move. It's DNA on legs. Right? Here's another look at bacteria really quick. Showcasing all those typical prokaryotic structures that it has. So circular DNA in the middle, no actual nucleus. Cell wall and cell membrane. The cell wall is made up of a substance called peptidoglycan, which is a type of protein. They tend to be motile because they have either cilia or flagella. In this case, this would be your flagella. They have pili, these little branches off of their cell membrane for attachment so they can hold on to each other using their pili. And then they have a capsule that helps to form biofilms, and it's just a protective layer for the actual bacteria. Bacteria are highly useful to us. We use it in our food industry to make things like yogurt and sour cream and milk and cheese. We use it in biotechnology to make medicines, and we've talked in depth about making insulin from bacteria. Personally, our bodies use it for help with digestion, like the E. coli in our gut. We use it to convert nitrogen into a usable form through nitrogen-fixing bacteria. Mostly plants do this. So they're very, very helpful to us. So this was just a little blip on viruses versus bacteria. Hope it wasn't too painful, and I'll see you guys next week. Bye.